Hello, everyone. My name is Andrew, and I'm here to present the presentation called hyaluronan structures synthesized by smooth muscle cells in response to hyperglycemia-induced monocyte adhesion. So basically, just to start things off, I just wanted to take the time to explain that our study is based in determining the role of monocyte adhesive HA matrices formed by airway smooth muscle cells in diabetes-induced lung injuries, which is kind of just the whole point of our presentation. So first, for this slide, called hyperglycemia-induced formation of monocyte adhesive hyaluronan matrix, which is trying to show that under the normal glucose concentration, HA is synthesized on the cell surface by HA synthase to form the surface coats, as shown in the picture there. And this cell surface HA cannot bind to monocytes or macrophages on their own. But then when we add this next picture in, what we found was that under hyperglycemia conditions, the glucose stress ends up elevating the cytosolic substrates, being the UDP glucuronate and UDP-N acetylglucosamine, to initiate synthesis of HA, which then deposits this high molecular weight polyanionic molecule in the intracellular compartments. This ends up initiating an ER stress that drives a unique autophagy in inducing the formation of extracellular monocyte adhesive HA matrix. This HA matrix, as shown in the image, can then bind to the monocyte/macrophages, and this interaction mediates the inflammatory responses that leads to diabetes-induced tissue injuries. And so in this next slide, which we labeled hyaluronan structure synthesized by smooth muscle cells in response to hyperglycemia-induced monocyte adhesion, this slide was basically just to show that in order to determine the formation of monocyte adhesive HA matrix by the smooth muscle cells in response to hyperglycemic conditions / serum starved, we had near confluent smooth muscle cells that were cultured for 72 hours in medium with 10% FBS in normal conditions, as well as high glucose concentrations, as well as, or even in normal glucose concentrations with 20 millimolars of mannitol. This monocyte interaction with cultured smooth muscle cells was then assayed by U937 cells. And what we ended up finding that we ended up labeling this bar graph was that the number of monocytes bound to the smooth muscle cells cell cultures were determined by ImageJ software. And the actual result was that high glucose induced a robust monocyte adhesion that can then be diminished by hyaluronidase treatments, suggesting that this is an HA-mediated adhesion. Further, the presence of serum ended up being required for this high glucose induction through the monocyte adhesion to the smooth muscle cell cultures. And in this image, which we label the confocal analysis of HC transfer on hyaluronan matrix in SMCs in response to hyperglycemic glucose with or without heparin. So basically, what we have here is that in order to determine whether there was a heavy chain transfer to this adhesive HA matrix, serum starved cells were cultured for 72 hours in medium with 7.5% FBS and 2.5% mouse serum containing different glucose concentrations. This was then stained for HA, which is labeled in the green, as shown in the images, as well as heavy chains, which is labeled in the red. What we found was that there were large cable HA structures observed in the high glucose concentrations showed in panels D through F, and in high glucose plus heparin-treated cultures shown in panels G through I. This, however, was not found in the low glucose cultures labeled panels A through C. In order to observe the red staining closely, green staining signals were decreased in panels E, F, H, and I, and the red staining signals became more dominant. Thus, it was clear that there were red stained coalesced heavy chain structures, as indicated by the arrows along the green HA structures. Interestingly, along the continuous hyaluronan cable structures, the red stained coalesced heavy chain structures were not forming the yellow signals, suggesting that the red staining was not colocalized spatially with the green HA signals. These results suggest that the modification of HA structures by heavy chain transfer was not occurring evenly along the cable structures. So in summary, our study showed that airway smooth muscle cells activated monocyte adhesion in response to hyperglycemic glucose concentrations by significantly increasing the HA on the cell matrix. The heavy chain modified HA structures were then observed on the monocyte adhesive HA cables. And altogether, these results support the hypothesis that hyperglycemia in airway smooth muscle cells induces synthesis of an HA matrix that recruits inflammatory cells and establishes a chronic inflammatory process and fibrosis that leads to diabetic lung injuries. And yeah, that more or less summarizes my presentation. Thank you everyone for coming.